Hello all, thanks for having me at TESS 2021. I'm Isabella Goulart, and I'm going to be presenting today the evaluation of Poincillonioidal relationships based on phylogenetic morphometrics. Poincillonioidal are Cryptogera adapted to marine environments, and these adaptations are forelimbs modified in fins, salt glands which allow them to drink salt water without dehydrating, a tendency to develop a secondary palate, and a hydrodynamic carapace. This phylogeny is from Gentry and collaborators 2019, and we are using it as a reference throughout this work. Poincillonioidal, according to Joyce and collaborators 2021, can be defined as the largest crown group containing Cheiloniamidas and Nemocheilus coriacea, but not Chelidra serpentina, Testudogreca, Trionyx triunguis, or Cynosternus copioides. So it contains the clades Democheilidae and Poincilloniidae. The Cretaceous composition of this group is uncertain, and it can also contain distinct groups Protostegidae, Toxocheilus latyremis, and Ctenocheilidae. The objective of the study was to do a phylogenetic inference of Poincillonioidal using multiple morphogeometric characters. But why is it different from previous attempts? Geometric morphometrics are very precise and less prone to subjectivity, since the lemma placement follows specific anatomical descriptions and are subjected to error testing. Of course, ideally, we should run the analysis with and without the molecular scaffold, but we have time limitations for this project and decided to test the phylogenetic relationships of Poincillonioidal without a molecular backbone. And we are using a different source of evidence from the ones previously used, that were traditional characters and molecular. And some terminal taxa cannot be well coded using traditional morphological characters, so Landmark can help to provide a tested evidence for the placement of this taxa. The in-group is formed by 31 species of Poincillonioidal, and a consensus configuration was used for species with more than one specimen sampled. The out-group is Chelidra serpentina, since Chelidrida is sister group to Poincillonioidal inside Americaelidia. The difference between traditional morphometrics and geometric morphometrics is to be able to work with shape. The set of landmarks, called a landmark configuration, represent the shape of a structure. But what is shape? Shape is the information we have left when we take away the effects of size, position, and orientation. Let's see an example with stars. Suppose we want to compare the shape of the stars. In this project, we use 2D images, so we first collect 2D images of the structures we want to study. Second, we define the landmarks that will suit the question we want to answer. We end up with a configuration that represents the shape of the structure. Then the centroid is calculated, which is the average point of all landmarks in the configuration. But the shapes are not comparable yet, not before the Procrucius operations, which will remove all non-biological information. The first one is the translation of all centroids to the origin. The second is to eliminate the effects of rotation in the images, and it is achieved by least squares. And the last one scales all configurations to a centroid size equal to 1. With biological structures, it's a little bit more complex, but you get the idea. We start with a 2D image, digitize landmarks on it, and end up with an aligned configuration of landmarks. For that, we use the TPS series of softwares by James Rolfe. We repeated the process for each of the 11 configurations using only the right side for symmetrical structures. Then we submitted them to error testing, and then a relative warps analysis was made to identify patterns of change in shape in each configuration. Phylogenetic morphometrics is a method proposed by Catalan and collaborators in 2010. This is a phylogenetic analysis using landmark data under a parsimony framework. TNT 1.5 already has the settings to work with landmark data. The goal of this method is finding for each landmark the ancestral position that minimizes the Euclidean distances between hypothetical ancestors and descendants along the tree, much like the approximations of character states in traditional parsimony. The landmarks have already been aligned by Procrucius operations, but in this point the landmarks are realigned to maximize the similarities between landmark positions that can be accounted for common ancestry. And why is that? Because Procrucius operations are needed to eliminate useless informations, like where in the plan the picture is digitized, or the scale of the picture, or the rotation of the specimen when the picture was taken. In this next step, the alignment is to minimize the distances between hypothetical ancestors and descendants in order to establish the cost of each transformation. The most parsimonious tree is the one with the minimum cost, summing up all these transformations. We combined our matrix of landmark data with a matrix of traditional morphological characters, and these two matrices were weighted so their weight would be equal, so they will have the same influence in the results. The traditional matrix from Gentry Collaborators 2019 was the most recent at the time and included matrices of marine turtles with a lot of fossil clades and some global matrices as well. Since we are only testing internal relationships and there is a computational limit of data that can be processed, we limited our sample to only Panchelonioid species. Each configuration was accounted as a character to avoid character dependence. The landmark matrix was composed of 11 morphogeometric characters, representing distinct osteological structure, and they were treated as continuous characters. Resistant fit theta row analysis is less sensitive to outliers, keeps the change restricted to the lemma set where it happened instead of spreading the change across the entire configuration like Procrustes does. And pairwise comparisons were made with Natato depressus because it has no missing characters. We made a traditional search, then another traditional search with the trees stored in RAM from the first one, and then a new technology ratchet. And Bremer supports were calculated for all global trees. We first run analysis with each configuration separately and then with sets of configurations, which are configurations that probably could evolve, such as pelvic and scapular girdles and shell, for example, to see phylogenetic signal of each structure independently and how they relate to one another. This is our main analysis, including landmark and traditional characters with equal influence on the tree, aligning with Natato depressus. The second not using character weighting, so there will be 11 landmark characters against 347 traditional characters, but will significantly lower the influence of the morphogeometrical characters. The third, excluding the ventral view of the skull, which is believed to be the most influenced by ecological factors. And the last one, aligning with Leptochelis olivacea to see if the alignment affects the analysis. This is our preferred topology from the first analysis. The first divergent clade in Panchelonioida is Ctenochelys procax. This species was described as Toxochelys procax by Hay in 1905, based on a fragmented skull. In 1953, Zongel changed it to Ctenochelys procax. And in 1997, Hirayama synonymized Toxochelys procax. Many others took Ctenochelys stenopus. And we decided to test this fossil as Ctenochelys procax along with fossils of Ctenochelys stenopus and Ctenochelys acris. In our preliminary analysis, it was recovered in the first divergence node of Panchelonioida or among Ctenochelys, but never in a monophyletic clade with Ctenochelys stenopus and acris. We are not able to find any other phylogenetic analysis using this specific fossil. Only Ctenochelys, generally speaking, or Ctenochelys stenopus or Ctenochelys acris. It shows no clear affinity with any genera, and the diagnosis of the species is based only on some vague cranial characters and on subjective comparisons with Toxochelys latyremus. So until we have more information on this material, we suggest treating this fossil as Incertasedis and consider the identification as Ctenochelys procax as provisory. Let's talk about Durophagous stem chelonids previously known as Osteopygidae. Osteopygidae, as we know, is polyphyletic, and they were characterized by the extensive secondary palate. Parham and Pienson 2010 proposed to keep the genus Erchilinesia to Erchilinesia goceleti and meridionalis and change Erchilinesia planimentum to Glossochelys because they lack the dorsally directed orbits of Phylloclastis and the elongated secondary palate of Erchilinesia. We did not find a dorsal view of Phylloclastis, but the orbits of Erchilinesia planimentum are more dorsal than those of most Cetodons. And when it comes to palate, is as extensive of that of Phylloclastis or even more. In every analysis where the two appear, they have an exclusive common ancestor, and with one of the greatest supports. Furthermore, the greatest Bremen support of all was in the group of the two Echinoneses, so we suggest to keep the genus for Echinonesa planimentum. And here's why we support Euclastes violandi as a Ctenochelide. In this phylogeny from Hirayama 1998, we have Osteopedes together with Echinonese in a polytomy with Ctenochelys and all Panchelonidae in the sample. The problem is that this is Osteopedes, not necessarily Imaginatus, and even if it was, the fossil was later proved to be a chimera. In Williams and Brown 2017, Euclastes is a wild card and appears in 8 possible positions, but they believe the divergence of this clade must have been in the middle of the companion because of the fossil record. Scavezzoni and Fischer were the only ones who specifically used Euclastes violandi, but they fell in the middle of a polytomy with all the Chelonidae. Euclastes in this work is associated with Ctenochelide in all trees, except ventral view of the skull and the one with the true views of the skull plus jaw, in which it's associated with Echinonese, probably due to the similarity of the palate, which may be convergence. So we suggest the Euclastes violandi be treated as a Ctenochelide. Careta and Lepidochelys olivacea are cosmopolitan and hybridize frequently. Camp not so much, since it's already rarer and restricted to the Gulf of Mexico region. Our results strongly support Caretini. As for Chelonini, we could not find any morphological evidence, only molecular. We reanalyzed Gentry and Collaborations 2019 dataset without a molecular backbone, and the main changes were in the extant species. The point is that the phylogenetic hypothesis of extant turtles is only being tested with molecular data, since all morphological ones use a molecular backbone and let fossils find their ways into an unchangeable tree. And we are accepting molecular hypotheses as true. For example, they are much more susceptible to long branch attractions than morphological data. And maybe the mess we are seeing here is not really a mess, but one possibility for the evolution of sea turtles. And what about Protosteidae in Crown Colonioides? Protosteidae inside Crown Colonioides pushes the origin of this clade far back and implies an extensive ghost lineage to Chelonidae. But why is that? Perhaps a convergence of specialization to a pelagic lifestyle? Everson Benson, 2019, still recovered Protosteidae within Chelonioides even after deleting characters associated with a pelagic lifestyle. Other important point to mention is that Santanachelys gaffnei is often used as sole representative of Protosteidae in phylogenetic analysis. Ocepetalon has characteristics of both Protosteidae and Demochelidae. In our analysis, it was recovered inside Protosteidae, like in Evers and Benson 2019, Evers and Collaborations 2019, and Gentry and Collaborations 2019. Mesodermochelys undulatus is the only Cretaceous Demochelidae in this analysis. In all global tree, it was recovered as sister group to Ctenochelide, associated with other Mesozoic turtles. Despite that, we do not defend the placement of Mesodermochelys undulatus, since we are not able to include a lot of morphological information of this species in this work to be able to contradict the strong evidence of the species as a stem Dermochelide. Protosteidae was recovered as part of Chelonioides crown group, and the recovery Caretini with a strong sport. And in this analysis, it was not possible to find an exclusive common ancestor to Panchelonidae. It was pointed out by one of our revisers that including more known marine species could help to improve the resolution at the base, so this will be our next step. Ctenochelys procax, that we believe should be treated as Insecticedis, the tribe Chelonini was not recovered. Ocepechelys buyai showed more affinity with Protosteidae than with Demochelidae, and Euclastes violandi recovered in a monophyletic clade with Ctenochelys. Our many thanks to our friends, colleagues, and family for support and counsel, especially to Santiago Catalano and Gustavo Oliveira for their counseling and revision, Leonardo Berig for giving us access to the cluster at his lab, James Rolfe and Willy Hennig Society for making the softwares we use freely available, Siriusha Evers for lending us some of his pictures, and the Univali team for giving us access to their collection. And thanks to CAPES for funding this project. Thanks very much. Muchas gracias. Muito obrigada. Our main references are in the following slides, and since the presentation is recorded, you can pause the video to read them. Bye bye, thank you.